Chapter 14 introduces us to the concepts of genetics, specifically Mendelian genetics. Genetics is the study of inheritance, of how genetic characteristics are passed from parent to offspring. Classical genetics is also called Mendelian genetics, and it was discovered by this guy here, Gregor Mendel, who was a monk, and he did experiments with pea plants, with garden peas, and one of them is shown on this picture over here. Mendel's experimental system was the garden pea. The Latin name is Pisum sativum. And as any model system, it had certain characteristics that made it a very good organism to use to study in general. So it was inexpensive, it grew relatively quickly, so he could observe a generation each year if he grew it outside. It was available already in many well-established varieties, so he could choose from seeds that would give him plants with purple flowers or white flowers, tall or short plants, etc. And it was easy to distinguish physically between these different traits. In other words, the tall plant would be much taller than the short plant. There wouldn't be any intermediate heights. The flower color was either purple or white, and so forth. So you could tell the differences between the varieties, between the traits within a variety very well. And then also, Mendel was able to control the fertilization of the plant, so he was able to breed those plants exactly the way he wanted to. He could either allow the plant to self-fertilize, which is a specific characteristic of that plant, or he could crossbreed them himself. Let's distinguish between characters and traits. A character is a feature that can be inherited that will vary from one individual to another, such as the height. In the case of the pea plants, they were either tall, about 2 meters tall, or short, about half a meter. The color flower, the color of the peas themselves, the texture of the peas. So all of those are characters. A trait is a version of a character. So within height, within the character height, you can have tall height and short height. Within flower color, you can have white or purple. Within pea color, you can have yellow or green peas. And within pea texture, you can have wrinkled peas or smooth, or also called round peas. When Mendel was doing his experiments with these plants, he would select characters that had traits that could be very easily distinguished from each other. So tall plants compared with short plants, round peas compared with wrinkled peas, yellow peas compared with green peas, and so forth. In the next couple of slides, we're going to take a look at some of these characteristics that the garden peas had. So here we have purple flowers versus white flowers. And another was the position of the flower. The flower position can be axial or terminal. Axial means that the flower is going to occur or is going to grow between where the leaf attaches and the stem, or the axil. So the flower would be here. That's an axial flower. And then a terminal flower is one that grows from the end or the terminus of the stem at the top. The color of the seeds or the peas, they could be yellow or green. The shape of the pea could vary. It could be round or wrinkled. And the shape of the pod varied. It could be inflated like this. It would look smooth and you wouldn't be able to see the peas growing inside. Or constricted. The constricted would look almost as if it were shrink wrapped around the peas, and you could see the shape of the peas growing inside. The pods themselves also varied in color. They could be either green or yellow. And the length of the stem of the plant could be tall, so the plant would be about 2 meters tall, or dwarf or short, and then the plant would maybe be half a meter tall. And each of these characteristics were independent of each other. So you could have round shape, and it could be green or yellow. So you could have round yellow, round green, and you can have inflated pods with round peas or wrinkled peas, or green peas that are round, or green peas that are wrinkled, or yellow peas that are round and wrinkled. You get the point. It's always nice to see an actual picture. So here we have peas with purple flowers and peas with white flowers. On the left side, we have inflated pods, and they happen to be green. And inside those inflated pods, there are smooth green peas. There could have been wrinkled peas in there too, and this particular variety is smooth. On the picture on the right, the pods are yellow and constricted. And those yellow constricted pods, there are green peas growing inside, and the green peas are wrinkled peas. So keep in mind, these peas that are wrinkled are not wrinkled because they're old or dried out. This is how they develop and how they come out of the pod. So it isn't so that the round peas, that these, if they dry out, that they turn like those. In fact, I have a bag of these round peas that I keep in my office. I've had it for several years, and they remain round. So they never become wrinkled like these. Mendel was able to do a lot of things with his pea plants because of a certain characteristic, which was that the pea plants that he was using are hermaphroditic, which means that in a single plant, in a single flower, you would have both male and female sexes. That is fairly common among plants. And here in this picture, it shows you a general representation of such a flower. And the male parts are over here, an anther that is sitting on a filament. And in the anther, the pollen grains will be made. And those contain the male gametes. The female part of this flower is this stigma that is sitting on a style, and it leads to an ovary over here. And then what normally happens is that the pollen from another flower will land on the stigma of this flower, and then it germinates, the pollen grain germinates, and a pollen tube grows through the style and goes to the ovary and transports the sperm cells to that ovary. Being hermaphroditic, then, this flower can produce both sperm cells and egg cells. The egg cells are produced in the ovary, and the sperm cells are produced in the anthers. We didn't talk about this term, monoecious. Monoecious refers to that both sexes are in a single flower. Monoecious means one house. Plants that have the two sexes separately are dioecious. A couple of examples of dioecious plants are corn and papaya. Hibiscus is monoecious. Here are the anthers, and you can see the yellow pollen that they make. And then the stigma are these little red structures. And what usually happens is that a bee collects pollen from one flower and then brings it to another flower so that you can have fertilization from different flowers. To avoid self-fertilization, in these flowers usually the stigma and the pollen mature at different times. So even if pollen were to land on the stigma might not be receptive to it. Another unusual feature of Pisum sativum, Mendel's pea plants, was that they self-fertilized. Usually when we have sexual reproduction, the idea is to get genetic variability by having two individuals contribute to the next generation. But Mendel's pea plants, as you can see here, the flower is closed, and at the time when the pollen is going to pollinate and meet the stigma, the flower is completely closed, so they tend to fertilize themselves. Notice that self-fertilization is not the same as asexual reproduction. In other words, meiosis still occurs. Meiosis makes the pollen and it makes the ovules in the plant, and then you have fertilization. It's just that the fertilization is from both of the gametes are made by one individual. In the last slide, we looked at an unusual feature of these pea plants in that the flowers are closed, and that usually the plants, if left to themselves, they will self-fertilize. Now Mendel sometimes would outbreed them, he would crossbreed them, and that is shown in this illustration. He would take one plant, and say this plant with the purple flowers, and he would take a pair of scissors and snip off the anthers of that plant. Now remember that these plants have both sexes on one flower. If you snip off the anthers, what are you doing? You're taking off the male parts. So what is left of that plant? This plant then becomes female. So the purple flowered plant is going to become just female. And then he takes a little brush, and with the brush he takes pollen from a white flowered plant, from this one, and he transfers the pollen from the white flowered plant to the purple flowered plant. And remember, the purple flowered plant doesn't have its own pollen anymore. It's a female flower now, so it is going to receive the pollen from the white flowered plant. In what is called a reciprocal cross, he does it the other way around. In other words, he would snip off the pollen, the anthers from the white flowered plant and have it become the female plant and deliver pollen from the purple flowered plant to the white flowered plant. So when he did these crosses with purple and white, and then he would wait for the peas to form, and then he would plant the peas and he would check what color are the flowers of the offspring when he did these crosses between purple colored and white colored flowered plants. He noticed that the flowers were always all purple, regardless of how he did it, whether the mother was the purple flower or the father was the purple flower. The flowers were always purple in that first generation, right after the cross. Before we go on, let's review a couple of definitions that we're going to be using in the rest of the chapter. We already saw a few of these. A character is a feature that's going to vary between two different individuals. So it could be the height, the color of the peas, the color of the flowers, the texture of the pods, the texture of the peas, the location of the flowers, etc. A trait refers to the variations within a single character. So if we look at the character of height, then the traits are tall and dwarf. If we look at the character of flower color, the traits are purple and white. The character of pea color gives us the traits green and yellow. Pea texture gives us smooth and wrinkled. When you cross or crossbreed a plant, you're going to be mating two individuals that have different traits. Like when Mendel was crossing the purple flowered plant with the white flowered plant, or if you do a crossing between a tall plant with a short plant. Notice that we're just mating individuals that display different traits of the same character. So we are comparing two traits of the same character. So purple versus white from the flower color character, green versus yellow from the pea color character. And then a reciprocal cross is actually two different crosses in which you're going to reverse the male and the female. In cross number 1, say the purple flower is the male and the white flower is the female. In cross number 2, the purple flower is the female and the white is the male, and so forth. And then a true breeding strain is a strain in which you always get the same traits, the same results when you breed among them. So if you have, say, a strain of purple flowered pea plants, if you cross them among themselves, these purple flowered pea plants, the flower of the offspring, generation after generation, they're going to be 100% purple. Same thing with a white true breeding strain. They're always going to have white flowers. Dwarf, tall, green peas, yellow peas, constricted pods, inflated pods, etc. Each of them was available as a true breeding strain. The parental generation are the plants in which the cross occurred, the original cross. So as when Mendel was crossing the purple flowered plant with the white flowered plant, those would be the parental generation. The first filial or F1 generation are the children or offspring of the P generation, of the parental generation. Depending on what characteristic you're looking for, it takes a different amount of time to get the results. If you were looking for whether the peas are going to be green or yellow or wrinkled or smooth, then when you do that cross you with the pollen and the anther, you just have to wait for the peas to form on the female plant, and then you have your results. But if you're looking at height of the plant, then you have to take the peas that form and plant them, wait till the plant grows to maturity, and then you can measure and see what the heights are. Or if you're looking for the flower color, you have to wait until they flower, until the children flower, and then you can see what the characteristics are. Then the second filial generation is the grandchildren of the parental, or the children of the first filial generation. And then finally, a hybrid is the offspring, is the child of two individuals from different strains. In other words, individuals that are showing different traits. So a hybrid between the white flowered and purple flowered plant, or between a plant that's tall and one that is short. Remember that cross in which Mendel would cross a white flowered plant with a purple flowered plant. And then when he looked at the F1, the first filial generation, regardless of which color had been the male parent and which color had been the female parent, he always got the result that all the members of the first filial generation had purple flowers. When he saw that, he called the purple trait dominant. And the other, the white trait, the one that you did not see in the first filial generation, he called that one recessive. In the same way, the pod shape, the inflated shape, is dominant over the constricted pod shape. So if you were to do a cross in which you had true breeding inflated pod, a strain of true breeding inflated pods, and a strain of true breeding constricted pods, and you crossbred them, the F1 would be all with inflated pods. If you had a strain of true breeding dwarf plants and crossbred them to a strain of true breeding tall plants, all of the F1 would be tall. So tall height is dominant over dwarf height, and dwarf height is recessive to tall height. Part of Mendel's genius is that he didn't do his experiments for only one generation. He took it another generation further. And remember that this means that he was maintaining the notes and doing this experiment for 2 years, because it took 1 year for each generation. So in the first mating, he had crossed a true breeding purple strain with a true breeding white strain, and he noticed that all of the members of the F1 had purple color. They were hybrids because they had one purple parent and one white parent, but all of them looked purple. When he then allowed these purple hybrid plants to self-pollinate, remember, in this case, he didn't have to do anything. He could just allow these plants to fertilize themselves because of their unusual structure. Then he would find that in the F2 generation, In the second generation, 3/4 of the plants had purple flowers and 1/4 had white flowers. And what we see here on the picture, they're even giving us the actual data that he obtained. He counted 705 purple flowered plants and 224 white flowered plants. He distilled this to a ratio 75% to 25%. In other words, 3/4 is 75%, 1/4 is 25%, 75% to 25% is a 3-to-1 ratio. So the ratio of the F2 generation for this type of cross is going to be 3-to-1. And he found that this ratio was repeated. It wasn't only with the purple versus white, but if he did it with tall versus short, or inflated pots versus constricted pots, etc., that ratio would hold. Now we're going to look at Mendel's results, applying what we know about meiosis and about sexual reproduction to the ratios that Mendel obtained with his crosses. Let's talk about two important terms in genetics, genotype and phenotype. In this slide, we'll be taking a look at the genotype. The genotype of an organism is its genetic inheritance, its genetic constitution. As we know, the genes contain information that codes for proteins, and that information is found in the sequence of the nucleotides. The genotype refers also to what that individual inherited from its parents. In a diploid cell, the genotype consists of 2 copies of every gene, 1 copy on each chromosome that the individual inherited from its parents. If the 2 copies of that gene are the same allele, then the individual is said to be homozygous for that characteristic. If the individual has 2 different alleles of that gene, then the individual is heterozygous. And then the genotype of an individual is going to determine that individual's phenotype. And let's take a look at the next slide to see what the phenotype is. The phenotype is defined as the part of the genotype that we can somehow detect, either by what we can see or what we can measure in some way. The one that is detectable visually is the most obvious and the easy one to determine. Things like whether a plant has purple flowers or white flowers, whether the peas are green or yellow, if the pods are inflated or constricted. Those are visual characteristics that you can look at and you can determine the phenotype of the individual. So the phenotype of that plant is purple flowers with green peas, tall height, etc. But it isn't limited to what you can see with the naked eye. For example, a blood type is also a phenotype, and you can determine that by doing experiments to see which kinds of cells clump with each other. It isn't necessarily directly visible. The functioning or lack of functioning of a protein, for example, if an individual can digest lactose, that is also part of a phenotype. In general, a phenotype can be dominant or recessive. In the case of the flower color, the purple color is a dominant characteristic, and white flower color is recessive. If an individual is heterozygous, Remember in the last slide we talked about that if an individual has two different alleles of a gene, it is heterozygous. So in a heterozygous individual, the one that is detectable in the phenotype, if there's only one that can be detected, it's called a dominant trait. And then the trait that is not detectable, the trait that is hidden, is the recessive trait. So our genes have the information that codes for the proteins and for our physical characteristics. These genes, all our genes, are located on our chromosomes, and here we see a pair of chromosomes. We're just looking at chromosomes with one chromatid each, but it would be the same if they had two chromatids. So this is a homologous pair, meaning that one of the members of this pair comes from the mother of the individual and the other one comes from the father. In this particular individual, we're looking at the characteristic of flower color, and we see that the individual is heterozygous for flower color because it has one allele that determines purple flowers and another allele that determines white flowers. Remember that the alleles are the ones that are going to determine the traits. And alleles are two versions of the same gene, but they will differ in the sequence of their nucleotides, and usually it is a small difference. In the previous slide, we saw that each gene is located in a specific part of a chromosome. So each gene has a locus on our genome, and that we have two copies of each gene. In a true breeding trait, let's say if you had in Mendel's piece plants that are tall and that have been tall for generations, and whenever you breed them with each other, the offspring are always tall, that is true breeding. In those individuals, both copies, both alleles for the height characteristic are identical, and that individual is then homozygous at that locus. If the individual is a hybrid, it means that it has two different alleles. The two copies of that gene are different, and that is also known as being heterozygous. And remember that if you have a heterozygous individual, the two alleles may be expressed differently in the phenotype, and only the dominant allele will be visible. If you have a cross breeding, so let's say in the example that we talked about earlier, you had these tall plants, true breeding tall plants. If you cross one of those plants with a true breeding short plant, in other words, the pedigree of that short plant is that it comes from a line where all the plants were short, and if you cross them to each other, all the offspring are short. If you now take a true breeding tall and you cross it with a true breeding short, you will see that all of the offspring of that cross are going to be tall. And that is a general rule, that the F1 offspring in such a cross breeding will all show the dominant allele only, which in this case is the tall allele. And it looks almost as if the short trait has disappeared. Like you mate the short plant with the tall plant, and there are absolutely no short plants in the offspring. But if you then allow those hybrids, those tall plants, to mate with each other, or you allow them to fertilize themselves, you will see that in the next generation, which is the F2 generation, the recessive characteristic will come back, the recessive trait will come back, and in the ratio of 3 to 1. And you'll see that 3/4 of the offspring will show the dominant trait and 1/4 of the offspring the recessive. In other words, out of 100, 75 would be tall and 25 would be short. Here's another example of such a cross. So in the P generation, the parental generation, we're looking at two true breeding strains. One that has purple flowers and the other one has white flowers. You cross them with each other, and what that means is that they're each going to form gametes, and those gametes are then going to come together in fertilization. So the purple flower makes gametes, and this represents the gametes made by the purple flower, a capital P, because purple is dominant over white in flower color. The white flower also makes gametes, and its gametes are all lowercase p. So let's say that the purple one is the male and the white one is the female. Then you've got sperm that determines purple and eggs that determine white. When you cross them together, you get a zygote, and that grows into an individual. And when it grows into an adult and it has its own flowers, all the flowers are purple. That is the phenotype of that individual. The individual is purple. It has purple flowers. The genotype is heterozygous. It has one allele that is the capital P for purple and another allele that has the lowercase p for white. Remember, it got the purple allele from the purple parent and it got the white allele from the white flowered parent. Now, when this individual, which represents the F1 generation, if you're going to allow the F1 generation to mate amongst themselves or allow the individuals to cross to fertilize themselves, then this individual is going to make its own gametes, and the gametes you can find out, you can represent them by separating the letters from each other. So it's going to make 50% capital P for purple and 50% lowercase p for white. So here you've got such crosses. They're giving us in the sperm you've got purple and white, and in eggs you also have got purple and white. And when you now cross them with each other, you find that the only way in which the offspring is going to have white flowers is if you cross a sperm that gives the white allele with an egg that also has the white allele, and that gives you this case here with little p, and that's going to be a white flower. The three other cases, if we look at the crosses, in this particular one here, you've got purple with purple, so that one is homozygous for purple. Purple with white, it is heterozygous, but because purple is dominant, the phenotype is going to be purple. Same here, heterozygous, but because purple is dominant, the phenotype is purple. So again, we arrive at that 3 to 1 ratio, 75% purple flowers, 25% white flowers. The little table that we saw in the previous slide is called a Punnett square. It is named after Reginald Punnett, who devised this way of writing the crosses so that you could keep track of the alleles that were being crossed. So it's basically a table, and on the top you're going to write down the gametes formed by one of the parents, in this case the male that contributes the sperm, and on the side, on the left side, you write the gametes contributed by the other parent, so the female who contributed the eggs. In this case, the both parents are heterozygotes, so they make 50% purple allele and 50% white. Their gametes contain 50% contain the purple allele and 50% the white allele, and the same for the eggs. And then we have that same cross that we saw earlier. You've got the different combinations, and each of these cells represents a zygote, represents the fusion between a sperm and an egg cell. And then that gives us this ratio, 3 purple flowered to 1 white flowered plant. Mendel formulated the law of segregation, in which he was referring to the fact that each individual has 2 copies of every gene, and those 2 copies separate in the process of the formation of the gametes. Now, Mendel didn't use those exact terms because the word gene had not been invented yet. That happened after he died. But he still realized that there were 2 copies. He called them factors. And he came to that conclusion, and in looking back at it with hindsight, he was exactly right. And what he observed was that the 2 alleles of each gene will be separated from each other during the process of meiosis, which is when the gametes are formed. They get separated or segregated into different gametes, and each gamete is going to have only 1 allele of every gene. So in the gametes, remember that during meiosis the number of chromosomes gets cut in half. What is happening there is that the 2 copies of each gene are getting separated from each other, and each daughter cell is going to end up with 1 copy of every gene. Then, when the process of fertilization occurs, the gametes are going to combine. 2 gametes, an egg and a sperm, combine to form a zygote. And the zygote is again going to have 2 alleles of each gene. So the normal number of chromosomes for the species is reconstituted during the process of fertilization. And then the ratios that we see, that 3 to 1 that Mendel observed and documented, is due to both the random combination of the gametes during fertilization and the interaction between the 2 different alleles, and the application of the dominance rule is going to give you the ratios that he observed and that holds across all of these crosses. So let's quickly review now the phenotype and the genotype that we already looked at earlier, but now we're going to add some information to it. The genotype of an organism is its genetic constitution, including all of its genes and alleles, and the allele is represented by a letter. Each allele is represented by 1 letter. So in a genotype, you will have 2 letters, a genotype that is just looking at 1 characteristic. So we're looking here at 3 different genotypes, this one, and this one. The genotype can be either homozygous or heterozygous, and if it is homozygous, it can be homozygous recessive, like this one in the middle, little t, or it can be homozygous dominant, like this last one here, big Y. And if it is heterozygous, then it has 1 copy of each of the two alleles. So big P, little p is a heterozygous genotype. This particular one, big P, little p, is purple flower, white flower genotype, which gives us a phenotype of purple flowers. Little t is two copies of the short allele, so that would be a short plant. And big Y is two copies of the yellow peas allele, so that plant would have yellow peas. The genotype also refers to the alleles an organism has with respect to just one gene, like we looked at in these three cases, or a few genes at a time. And then the phenotype of an organism is composed of the traits that we can observe. A tall plant, a plant with white flowers, a plant with constricted pods, etc. 3:1 ratio in the F2 cross that we saw earlier is interpreted as phenotype versus genotype. So in the phenotype, we're looking here at 3 purple flowers versus 1 white flower. But if we look at the genotypes behind those phenotypes, we see that in the upper one, that purple flowered plant, its genotype is big P, so it is homozygous dominant for the purple allele. Then there are 2 plants that also have purple flowers, and they look just like the one on top, but they are both heterozygous. They have one allele that determines purple flower, and the other allele determines white flowers, but it still is going to give a phenotype of purple, which looks exactly the same as the phenotype for the homozygous dominant plant. And finally, on the bottom, we've got one plant, the one in the 3:1 ratio, that has white flowers, and it is homozygous recessive. It has 2 copies of the white allele, 2 copies of the lowercase p. Notice another thing. The phenotypic ratio of 3:1 is composed or determined by a genotypic ratio of 1:2:1. And where does that come from? Well, the 3 of the phenotype, these 3 purple flowers, can be subdivided into 1 that is homozygous dominant and 2 that are heterozygous. So that 3 is composed of 1 and 2. And then this 1 of the phenotype is the same as the 1 in the genotype. So 3:1 in the phenotype is the same as 1:2:1 in the genotype. If you have a plant with purple flowers, you don't really know if it is a true breeding purple plant or if it is a hybrid purple plant, because you cannot tell from the phenotype. In other words, you don't know if its genotype is big P or big P, little p, right? If it's big P, then it is homozygous dominant and true breeding for the purple trait. If it is big P, little p, it is heterozygous and it can have both purple and white offspring. So a test cross is a special cross that we can do to be able to determine what the genotype of such an individual is, an individual that is displaying a dominant trait. In other words, like the question is, if the flowers are purple, How do you know if it's big P or big P little p? If the height is tall, is it big T or big T little t? If the individual is displaying a recessive phenotype, we know what the genotype is. In other words, in an individual displaying a recessive phenotype, what you see is what you get. Because if it has white flowers, you know the only way it can have white flowers is to have two alleles for white flowers. The only way it can be short is to have two alleles to determine short height. The only way it can have green peas is to have two alleles for green peas, etc. So what does the test cross entail? Well, it means that you have to cross this dominant phenotype individual to another individual that displays a recessive phenotype. And why is that? Because you already know the genotype of this individual with the recessive phenotype. You know that it has to be homozygous recessive. So in the case of the white flowered pea plant, you know its genotype is little p, little p. So if you cross it to this individual, then you will be able to predict what the genotype of the purple flowered individual is according to the ratios that you obtain in the offspring. In the recessive phenotype individual, what you see is what you get. So here we have that test cross and the two possible results that we can get. Remember that the homozygous recessive individual is a given. Its genotype, you know it is little p because it has white flowers. So that one is already known. The purple flowered individual can have two different genotypes. It can be either big P, as we see in this condition here on the left, or it can be big P, little p, as we can see here on the right. And each of those situations is going to give us a different outcome. Let's check on the left first. So big P crossed with little p. In each case, the zygotes are going to be big P, little p, right? Little p from the sperm, big P from the egg, little p from the sperm, big P from the egg in each of the four possible combinations. So it's going to give us an offspring that is 100% purple, regardless of how many offspring there are. If there are 100 offspring, it's going to be all of them purple. If there's 500, they're all going to be purple, etc. Now let's take a look at the other situation. Again, the homozygous recessive parent is a given, is known, and then the other parent is showing us now the other case in which you can have an individual that is phenotypically purple, but it is heterozygous or hybrid, so it produces 50% of gametes with the purple allele and 50% of gametes with the white allele. And when you cross those to each other, you're going to get the result that half of the offspring are going to have the purple color and the other half are going to have the white color, giving you a ratio of half purple and half white offspring, or a 1-to-1 ratio, 1-to-1 phenotypic ratio. In this slide, we recap the results that we saw in the previous slide. So we're looking at the two possible results. Either all the offspring are showing the dominant phenotype. In this case, all the plants have purple flowers of the offspring, or half the offspring have the recessive phenotype, white flowers, and the other half of the offspring have the dominant phenotype of purple flowers. Now remember, in a cross like this in pea plants, you're going to have several hundred offspring. So you basically, if you see that all 260 offspring have purple flowers, then you can be very confident that the purple flowered parent is homozygous for the purple allele. If you only have 3 offspring and they're purple, your confidence isn't as high because you simply don't have a statistically significant number of offspring yet. The crosses that we have discussed so far have been looking at only one trait in the organism. The same rules that apply to one trait apply to the whole organism. We're going to slightly expand our reach by looking at 2 traits at the same time. And the traits that are selected for this cross, looking at 2 traits, are the color of the piece, in which we are going to be comparing yellow versus green, and yellow is dominant over green, and the texture of the piece, where we compare round versus wrinkled, and round is dominant over wrinkled. Let's take a look at the first generation, the parental or P generation. In that parental generation, we're crossing an individual with the genotype big Y, big Y, big R, with another individual that has the genotype little y, little r. Those are the genotypes. The phenotype of the big Y, big R plant is yellow round peas. The phenotype of the little y, little r plant is green wrinkled peas. Both of these plants are true breeding. That means that each of them is homozygous for both the characteristics. The plant with yellow and smooth peas is homozygous for the yellow allele and also homozygous for the round allele. And the plant with the wrinkled green peas is homozygous for the green allele and also homozygous for the wrinkled allele. In preparation for the cross, the 2 parents are going to make their gametes. The only kind of gamete that the yellow round plant can make is big Y, big R. Right? In meiosis, you're going to separate the homologous chromosome pair, so you're going to take the big Y on one side and the big R on the other side, and that's going to give you big Y, big R. Any combination of Y and R is going to have big Y and big R. You can take this Y with that same R as before, and you still get big Y and big R. In the other parent, same thing. You get one Y and one R, whichever one you choose, it's going to be little y and little r. When you cross them together, then one represents a sperm cell and the other one represents an egg cell. The fertilization entails the fusion of the sperm and the egg, so the F1 generation, the children of these 2 parents, are going to have this genotype and phenotype combination. The genotype is big Y, big R, little r, because they got a big Y from the yellow parent and a little y from the green parent. They got a big R from the yellow parent and a little r from the green parent. So that's the genotype. Once you have the genotype, you apply Mendel's law of dominance and you determine the phenotype. You look at the two copies of the same character. So you've got big Y and little y. Big Y is for yellow peas, little y is for green peas. Yellow is dominant over green, so phenotype is going to be yellow. For the other character, big R is for round shape, little r is for wrinkled shape. Round is dominant over wrinkled, so the phenotype is going to be round. So that gives you an individual with yellow color in the piece and round or smooth shape. And all of the members of that F1 generation are going to have that same genotype and phenotype. As soon as you are starting to look at two characteristics at the same time, a very valid question arises, and that is, do the combinations of alleles that we see in the gametes of the parents, are those combinations going to be the same combinations that are already present in the parents, or can the alleles of the two different genes assort independently from each other? In other words, if you have a heterozygous parent, like in this case you have big Y, big R, little r, do the two Y's segregate independently from the two R's? Let's make the pencil not go away. So if you have big Y and big R and big Y and little r, is that a possible combination? Or is it so that big Y always is going to be together with big R, like it was in the grandparent, and little y always with little r? So the outcomes are going to be different. If assortment is independent, then this individual, big Y, big R, little r, is going to be able to make 4 different separate gametes. And we're going to see that more clearly in an upcoming slide. If they stay together, like they were in the grandparents or in the parents of this individual, then this combination, this genotype, would only be able to make these gametes, big Y, big R, and little y, little r, just like it inherited them from its parents. I'm going to give away the answer. The answer is that assortment is independent. So the Y's and the R's can separate independently from each other, and it is the first condition, it is the first situation that happens. The individual can make 4 different kinds of gametes. In this case, big Y, big R, big Y, little y, big R, and little y, little r. So this slide tells us again, assortment is independent. The two characteristics will assort and segregate independently of each other. So that gives 4 different gamete combinations for an individual of genotype big Y, big R, little r. It's going to give you those 4 different gametes. And when you cross 2 individuals with these characteristics, we have to make it no fade again. when you cross two individuals that have that genotype, then the combinations of zygotes, or the ratio of offspring, is going to be also a very typical ratio, very characteristic ratio, 9 to 3 to 3 to 1. The two parents, for this ratio, the two parents are both heterozygous, big Y, big R, little r, and this is therefore called a dihybrid cross. Both parents, di, are heterozygous or hybrids for both conditions. So this ratio of 9 to 3 to 3 to 1 will occur only in a dihybrid cross. And what do we mean with that ratio of 9 to 3 to 3 to 1? We mean that out of all the offspring, you have to now count what is all the offspring. Well, you have to add all of these up. 9 plus 3 is 12, plus 3 is 15, plus 1 is 16. So out of 16 possible types of offspring, 9 of them are going to have the two dominant phenotypic characteristics. So 9 are going to have yellow peas that are round. 3 are going to have one dominant and one recessive characteristic, so they're going to be yellow with wrinkled peas. Another 3 are also going to have one dominant and one recessive, but the reverse one, so they're going to have green peas that are round. And then finally, 1 out of 16 is going to display both recessive characteristics, so it's going to have green peas that are wrinkled. And we're going to see this in a picture in an upcoming slide. So here we are comparing again the two hypotheses of dependent assortment and independent assortment. Dependent assortment means that the combination of alleles in the gametes is the same as in the parents of the individual, so they are going to pass down exactly the alleles in the combination that they received from their own parents. So that would only give you big Y, big R, and little y, little r in the offspring. The other hypothesis is that assortment is independent, so the individual is mixing the alleles independently of each other, the characteristics independently of each other. So you get big Y, big R, big Y, little r, big R, and little y, little r. In other words, you are mixing and matching the characteristics in all possible combinations. What are the rules for these combinations? The only rules are that each gamete will have one Y and one R. That's the only thing. You have to make sure that you've got that you have one copy of each chromosome, and that is what is represented in that one Y and one R. On the top of the Punnett square, we're looking at one parent, and on the left side, we're looking at the other parent. This was a dihybrid cross, right? Both parents, each parent has this genotype, big Y, big R, little r, crossed with itself. So if I do that, crossed with, and then again, big Y, little y, it's hard to write with your finger, big R, little r. So that's the cross that we're looking at. And these over here that we already saw are the gametes that the two parents form. And inside, that we're looking at in here, in the table, are the different zygotes. And this is where we're going to generate the 9 to 3 to 3 to 1 ratio. notice that if you count them, there are 9 P's that are both yellow and round. Shall we count them? 1, 2, 3, 4, 5, 6, 7, 8, and 9. And then there are 3 that are going to be yellow and wrinkled. Let's make those a different color. So let's see. 1. You can't really distinguish that. Let's make it black. 1, 2, and 3. Another 3 are going to be green and smooth, and you can see them here. 1, 2, and 3. And finally, 1 is going to be green and wrinkled. And I used the blue one. 1 individual there. And that gives you the 9 to 3 to 3 to 1. In the case of the yellow and smooth individuals, notice that there are different genotypes. Like this one over here is big Y, big Y, big R, big R. This one is big Y, little y, big R, big R. 2 different genotypes, but they give the same phenotype, because when you apply the law of dominance, yellow is dominant over green and round is dominant over wrinkled. As far as Mendelian genetics is concerned, assortment is independent. When genes are located on different chromosomes, they assort independently. So this situation that we see on the left side is not going to happen. What makes possible independent assortment is the fact that genes that control different characters, in this case the color of the pea and the texture of the pea, are located on different chromosomes so that they can separate independently from each other. Let's take a look at the chromosomes in this case. Pea plants have 7 pairs of chromosomes, and they're showing us 2 pairs in this particular example. They're showing us a long pair of chromosomes that have the R characteristic, and the R characteristic stands for the texture of the piece, with capital R being round and lowercase r being wrinkled. So that's carried on the large pair of chromosomes. The small pair of chromosomes over here carries the color of the pea, so the character of the color. And big Y represents yellow peas, little y represents green peas. So the color is on a different chromosome pair as the texture. On the left side, we see an instance of meiosis in which the chromosome that controls the texture of the pea and the color of the pea, the two dominant alleles segregated to the left side, and the 2 recessive alleles segregated to the right side. So you've got, on one side, you've got big R and big Y, and on the other side you've got little r and little y. And look over here, you see when the chromosomes separate from each other, one daughter cell is going to get big R and big Y, and the other daughter cell is going to get little r, and little y. Now let's look at a completely different event of meiosis. So we're going to make a line here to symbolize these are two independent meiotic events. And in this case, the distribution of the chromosomes was different. So if you look down the line here in metaphase, you have the chromosome with the little r and the big Y segregating together, and the chromosome with the big R and the little y segregating together. And that's going to give us daughter cells. One of them is going to have the little r and the big Y together, and the other one is going to have the big R and the little y together. So that, when you look at them all together, now you can see you have all four combinations. You have big R, big Y, little r, little y, little r, big Y, and big R, little y, over multiple meiotic events. This picture shows us the whole process of meiosis with both divisions. So on each side there is one meiosis, and one side gives us the one combination that we talked about earlier. So you've got big R with big Y and little r with little y. And here you have the four daughter cells produced in that meiotic event. So you've got big Y and big R. Two of them have that, and two of them have little y and little r. Then, if you look at another possible meiotic event, you have the other combination. So here two daughter cells have big Y, little r, and these two daughter cells have little y, big R. Now we are looking at the meiotic production of that individual as a whole. So we're not looking at them just one meiosis and the other meiosis, but we're looking at the products of meiosis as meiosis as a process. You have now every one of these combinations. So this is why it tells us you've got a quarter of the gametes are going to have big Y, big R. A quarter is going to have little y, little r. Another quarter is going to have big Y, little r. And the last quarter has little y, big R. So you've got all four different allelic combinations in the gametes that are produced by an individual with a dihybrid genotype, an individual that is a heterozygote for two different characteristics. We saw in the previous slide that independent assortment is responsible for the fact that each parent in a dihybrid cross is going to give you this combination of gametes: a quarter big Y, big R, a quarter big Y, little r, a quarter little y, big R, and a quarter little y, little r. You've got the same for the mother, because what we looked at on the top were sperm, so those are the father, and here on the side are the gametes that the mother produces, the eggs. She also makes a quarter big Y, big R, a quarter little big Y, little r, a quarter little y, big R, and a quarter little y, little r. That combination of gametes is going to give us the 9:3:3:1 ratio, because you are going to now combine these gametes randomly in the process of fertilization. If we just take a few examples, let's combine this sperm with this egg. Where do you see that that result? Well, you follow the line down for the sperm, and you follow this line to the side for the egg, and where they cross is the zygote produced by that particular combination. So if you cross little y with little r from the father, with little y, big R from the mother, the combination you get from that is in the zygote little y, big R, little r. And that is going to be a green pea, because it's got little y, that is smooth, right? Because round, or it is round because round is dominant over wrinkled. And for each of those you have the combination. So when you've got all possible combinations, you're going to get that 9 of the offspring are going to display both dominant characteristics, in other words, both yellow and round. 3 are going to be yellow and wrinkled, that's 1 dominant and 1 recessive. Another 3 are going to be smooth and round, 1 dominant and 1 recessive. And 1, this guy over here, is going to have both recessive characteristics, wrinkled and green, which is little y, little r, little r. Of course, you also have to apply the rule of dominance to get the phenotypes in the offspring. So it's a combination of all 3 things: independent assortment when you make the gametes, then a random combination of those gametes, and then when you apply the rule of dominance in each case, then you're going to get that ratio. Remember, the ratio of 9:3:3:1 is a very specific ratio. It only occurs when both parents are heterozygous for both characteristics. Any other combination, let's say if one parent is heterozygous for both, but the other parent is homozygous for one and heterozygous for the other, is going to give you a different phenotypic ratio.